United States Department of Agriculture.
Okay, thank you very much for the invitation, and I hope to entertain for the next 20 minutes about home cage technologies. Okay, so we saw from 2022 a huge movement towards AI, so artificial intelligence, and specifically large language model. We know that ChatGPT has been launched, and now even Jenna passes the medical exam, and in one case was able to diagnose one disease with one person that could actually not been diagnosed by doctors. However, on the basis of all of this, we have digital data. So we have tons of data that are properly organized and are fed into these huge models to be able to reach what has been reached here. In animal model or in laboratory animals, it's very difficult to reach that because we don't have the structures yet in place. However, what we can do is start the process, and at some point we will be able to, and I'm pretty sure we will be able to reach that goal. So we need digital data. 24/7 system are being used now for digitalization, and I'll explain later to you what we intend as home cage monitoring systems. I will bring some example for how AI can help in the welfare, what we intend as a digital biomarker, as an example of the COVID-19 model. And last, building repositories, so building databases that can be then inquired, as I showed before with the ChatGPT, and what is needed in order to be able to have decentralized information, and each person, potentially, or scientist could inquire such databases. So typically, when we perform experiments, we move the animals from their home cage into experimental settings. Can be behavior, can be something different during the day, sometimes in the reverse Ladakh cycle, but that's what we typically do. And then if the experiment is not final, we move the animal back in the home cage. There's been trends in the last, I would say, 10 years to move some experiment back in the home cage because we can actually monitor the animals also in the night. You have better observation time and not just limited to, like, for instance, 5 minutes. And for long processes like age and environment play a huge role. You can better control these factors. And there are two big groups, one in Europe, Costi Time, and one in North America, that actually are working to define what home cage monitoring is and what are the methods that should be used for this in these technologies. Within the 3Rs collaborative, we brought a paper out in which we define two types of home cage monitoring. One is rack-based, which is leaving the animals where they live 90% of the time and bringing the technologies into it. The other one is like benchtop technologies. You see some pictures here, in which you literally move the animals from their home cage into a new cage. They can stay there for some days or weeks, and eventually they have to go back into the home cage. And there you can perform more complex behaviors, like what you see here with RFID systems. And there are some limitations on both sides. What we intend as a digital biomarker or human endpoint is bringing the technologies into the cage. So we can have a camera, RFID, electromagnetic fields, and essentially identify two states. One is the physiological states, in which there is some variation between day and night, which is physiological. And in this case, with a red line, when the animals become pathological, it's a lung cancer model. There is a reduction of the parameter, in this case, 50% of the activity is reduced. And if you give a drug or you do an intervention, the animals can go potentially back into the model before that is too late or the animals has to be terminated. So as an example, one system is the digital ventilated cages, in which we essentially, at the cage level, underneath, we have a sensor which senses the cage content with micro electromagnetic fields, 4 times a second. And then we can sense three things, essentially. One is the cage getting dirty because the animals urinate. Second is the water spillage of the water resistant bottles. And third are mice. And because these have three different dynamics, you can tell them apart very, very easily. So now I mentioned to you the Costi Time, which is the European Home Cage Monitoring Association. It is in the last year of work. Essentially, they ask a survey with 279 respondents. What are the factors for you to take home cage monitoring into consideration? And one of the negative factor is the financial aspect, so which is a pain, right? You need money to acquire these large systems, to let them run, to store the data, and all of the stuff. And now, consider one of the operational side, the cage change frequency. I don't know how much in your facilities you do cage change, but typically it's weekly or bi-weekly, and it's dictated by essentially one factor. Obviously, you can control better the animals, you can check them, but the other one is that there is one parameter which is defined, the ammonia, which is derived by the urination of the animal, in which 25 ppm of ammonia are considered unhealthy for animals. And this is based only on human studies. So there was exposure studies around on humans, and 25 ppm of ammonia was considered unhealthy for the animals as well. That's dictate the weekly or bi-weekly cage change in respect of the number of animals in the cage. Where there are a few papers that came out that essentially show that if you actually have 1, 2, 3 animals in the cage, like in 57 Black 6, and you extend the cage change, it does not cause any discomfort to the animals. There is no pathology occurring. So this 25 ppm is not dictated by any scientific finding. And so what you could do to help with the cost, you can adopt an AI-aided cage change in which you have experiment with 2, 3, 4, or 5 animals per cage. This is a facility in the US. You tell the system, this is our cage, not 30. At 7 days might be more or less 30 according to the number of animals in the cage. And then you reduce the number of animals, depends on the number of cage changes, depends on the number of animals in the cage by average 58% or 50%, depends on the facility and your sense of dirtiness of the cage. Therefore, the return of interest is in 3 years. So in 3 years, you start actually reducing the cost. And you also, in Europe, it's very important to have the CO2 footprint. So it becomes sustainable. It's halved within 1 year if you have 1,000 cages equipped with technologies like that. So already one of the pain points is reduced by such approach. But let's say that we don't care about the operation. I'm mostly interested about the welfare and the science. And so one of the questions that we always been asked is, what is the impact of the low or bad welfare or distress of the animals on the science output? And one of the things that typically happen in many facilities are 2 things. Fighting in male animals and how to deal with it. And second is, as you see at the bottom, repetitive behaviors that occur because the animals might be distressed because of many factors, and how to quantify them and how do they affect the output science in that case. So what we adopted was an AI approach. So we train a system with video. So we video monitor the cages. We tag different parameters, 10,000 videos with fighting and repetitive behaviors. And then we train the system, which is derived from our system, to say yes or no. Is that stereotypy, so repetitive behavior? Was that fighting or not? Or was it just negative? Was nothing, as confirming what you essentially see when you do your cage control during the day. So in 94% of the cases, it's negative. That means what the algorithm predicts is also what you see. But in other cases, you can actually have repetitive behaviors in 74% of the cases, or fighting, that you don't see because they're mostly happening during the night. And one group in University of Vienna in Austria asked the question, what happens if you have repetitive behaviors in the cage? So what they identify is the following. It's an arbitrary identification. So identify number of events in male and female mice. If they were 0 to 1, they identify as low stereotypies. 0 to 0.5 on average per day. 0.5 to 1 was medium, and above 1 events per day was high. And the question was, what happens if I put them in the open field? Open field is a test which can be used for anxiety, but also to measure the general locomotion of the animals. And essentially, what happened in female, there was not much of an effect, but in male mice, if you have animals with only 4 average stereotypies or repeated behaviors per day, you have an increased activity of all the animals in the cage. And if you give anxiolytic, like in this case, you will see exactly the same. So how do you know that is due to the anxiolytic effect and not to the repeated behaviors? And very similar, they did also a force swim test in which you put the animals in a jar with water. And if the animals are struggling or they're swimming, they're considered less depressed-like behavior. And if they're floating, they're considered more depressed. And again, in male with high, considered high, so 4 events per day, they swim more, they don't float. And again, if you give an antidepressant, which is what the force swim test is based upon, you will see exactly the same effect. Again, so if you know that the animals have a low welfare, you can predict or exclude such cases in order to be able to do not confound your results. Now moving to the science of things, if you work in infectious disease, you know that typically to identify the disease progression of the animal, you look at the daily body weight. And it's a problem for those that perform the daily body weight because it's risky, especially BSL-3 and 4 approaches. And sometimes the body weight might be not the best indicator of the animal welfare. So like in the case of the COVID model, we have several publications that came out with this humanized ACE2 receptor from Jackson. And they share several clinical features, but one that is not being shared across mice and human is the weight loss. And so one of the very first publications came out in 2020 was simply subjecting these mice to SARS-CoV-2. And what you can notice is that the body weight in SARS-CoV-2 treated animals only change after 5 days of the treatment. At day 7, the animal has to be stopped because they're very sick, and the experiment has to be finalized. We put the very same animal in normal cages, and we asked the question, what happens if you look at activity? What you see on the left side, you see the body weight change that starts to actually drop 1 day prior to the previous publication, but the activity is already reduced at day 1, which is actually resembling our fatigue that we have during infectious disease or during SARS-CoV-2 for those that had it. At day 4, it's already reduced by 90%. So we could actually say that we can complement well the original gold standard in vivo parameter with such new parameters. There are many more publications that I don't have to go through. If you want, there is the QR code to it, and you can scan it for a second and go and check such publication if you're interested into it. Last point I want to talk to you about today is about the usage of this data and how can we organize in order to actually not refine, but to reduce the number of animals in a publication. It's called virtual controls. So typically, we know that we have tons of clinical data, we publish them, and the significance of the meaning. So with the question we've been asked, why do over 90% of behavioral neuroscience results into fail in translation, and how can we fix it? It's one of the points that we've always been exposed to. And very recently, the NIH grants start demanding data repository for in vivo data. So when you write a grant, like an R24, you've been asked, where are you going to deposit your data? And there are several repositories in Europe and also in US. They started from genomics, and now they also go for in vivo data. But especially for the in vivo data, we don't have a consensus on how the data should be structured in order that if I, from Europe, ask a question as an experiment already been performed, I will find the information right off the gate. And so what we came out in 2024 was a publication in which we suggest a minimal set of information called metadata that could be used to actually tag the raw data in order to be able to find them in several repositories. So the idea is if you have several experiments, you will have certain subsets of information that are common to the different experiments, and therefore you can use the raw data associated with the tag data and inquire, was this information already being used? This can generate data sets of databases at the institute's level with standard structures that would allow you, like at Yale or other university or NIH, to inquire was like male being used for this type of study, female, genetically modified animals. This is just an example of one of the institutes that has been starting to use such a structure. The idea is that scientists become more reproducible, potentially efficient, and ethics, because not only the positive data will be published and will be deposited, but all the data theoretically. The biostatistician can be also more precise and replicable, and the regulatory bodies can become more transparent. And also there will be a reduction in the cost and potentially higher animal welfare. What could also potentially be used is for control animals, the data could be reused across different institutions, and you can better reconstruct. Instead of using 8 animals, you could use potentially 2 or 3. This is something that has been already started in the pharma. It has gone for 5 years, and it's been bringing results like reducing the number of animals used in research by 30%. And coming back to the original idea, potentially we can then ask in the chat, bring me the data of a 57 black 6 at baseline at age XYZ, and get the information we need. Very similar to the Jackson information or library that are out there, but with much, much more data, and finding also the data that are out there. And with that, I would like to thank you, and I'm open for questions.
USDA is an equal opportunity provider, employer, and lender.